I'm Dicao. The study is fast whole brain MRI imaging of dynamic susceptibility contrast changes in the CSF. Recently, lymphatic system was found in the brain as a complementary to the cardiovascular system. Main function of lymphatic system is clearance of waste in the brain. And there are two key components of lymphatic system in the brain. One is a brain-wide lymphatic pathway between arteries and veins, which is responsible for influx and the transportation of CSF. Another is membrane-based lymphatic vessels running alongside the major arteries and veins, which is responsible for CSF efflux. In this study, we aim to develop a method to measure the dynamic signal change in the cerebral lymphatic vessels. It's well known that the dural vessels don't have brain-blood barriers, so that the gap in vessels could get into perivascular space. And there are many gap-based MRI methods to image lymphatic vessels. Here is one of the studies that shows three main regions of lymphatic vessels in the brain, which are superior sagittal sinus, olfactory region, and meningeal arteries. However, most studies adopt inversion recovery-based sequences, such as FLAIR and black blood, which usually requires relatively long acquisition time, about 5 to 10 minutes, and may have partial volume effect. Visualization of the dynamics of these neural fluids could be one of the important ways to evaluate the lymphatic system. So we want to develop a higher temporal resolution sequence, achieving dynamic image of gap signal changes in the lymphatic vessels. A 3D turbo spin echo sequence was adopted and modified to achieving the following goals. First, long TE over 600 milliseconds was used to minimize signals from gray matter, white matter, and blood, and separate the CSF signal from the rest of the brain. Second, to avoid the extravascular effect from GAD, a spin echo-based sequence was adopted. Third, to get whole brain coverage with a temporal resolution of less than 10 seconds and a sub-millimeter spatial resolution, the sense was used to accelerate the acquisition. Last but not least, we did simulation to optimize sequence, achieving significant signal change in CSF before and after GAD injection. GAD shortens both T1 and T2 in the CSF, which affects the TSE signal oppositely. Therefore, two types of sequences were evaluated: T1 dominant sequence that furnish a positive contrast, and the T2 dominant sequence that furnish a negative contrast in CSF. First, to get T1 dominant contrast, we use shortest TE possible to minimize T2 effect. We need TE at least 600 milliseconds to suppress blood signal. Then we need to find optimal TR. Here is part of our simulation results. This same blue line represents CSF signal. The thick blue line represents signal of CSF with GAD, and red line represents blood signal and other tissue, which are all known. And the dark line represents contrast between pre and post GAD. We can see TR around 3000 milliseconds is optimal for positive contrast. For T2 dominant contrast, we need to use a long TR to minimize T1 effect. And we tried several values of TR and think TR equals to 9 seconds is optimal. Then we need to optimize TE. Here is result. We can see that the blood signal is also known. And the TE around 1400 milliseconds is optimal for negative contrast. We did further simulations of the signal contrast with and without GAD in CSF as a function of GAD concentration. Simulation results show that the T1 dominant sequence may have same contrast for different GAD concentration. Whereas in T2 dominant sequence, the absolute CSF contrast increases monotonically with concentration of GAD. When the concentration of GAD is uncertain in vivo, a biphasic relationship can be problematic, where a monotonic relationship may provide a method to test the concentration of the GAD. Therefore, the T2 dominant 3D TSC sequence was chosen for the subsequent scans. Then we did phantom study to validate our optimized sequence. We used 4 plastic tubes with solutions of water and GAD of different concentration to produce similar T1 and T2 of CSF with 0, 0.1, 0.5, and 1 millimole per liter GAD. Here is phantom results compared with simulation results. We can see that they agree with each other. And the T1 sequence has this biphasic problem, whereas the T2 sequence has monotonic decrease signal. Here is our protocol for 3T human study. We use MRA and SWI to identify big vessels. And we acquired FLAIR pre and post GAD injection, like previous study, to reproduce their results. And then is our dynamic 3D TSC sequence with 9 seconds per volume. We did 140 dynamic scans with GAD injected at 33 dynamic scans. For data analysis, we did motion correction for our 3D TSC data and did correlation for other images. And we use CNR larger than 1 as threshold to filter out the noise region in our 3D TSC data. Here is our typical results from human subject on 3T from regions around superior sagittal sinus, where lymphatic vessels were identified in previous studies. And the CSF signal change after the GAD injection was detected in FLAIR, similar to previous studies, as well as in our proposed 3D TSC sequence in current study. Here is average time course from 3D TSC. The star line indicate when the GAD was injected. Here are 4 parametric maps extracted from this time course. The T on side means the time point when signals start to decrease. T peak means the time point when signal comes to steady state. Delta S over S means relative contrast. And the concentration of the GAD can be calculated from this relative contrast. Here are results from 2 other regions. This is the middle meningeal artery region. And this is the olfactory region. Here is quantitative results showing mean and standard deviation from 8 subjects of 3 regions curve. Our on-site time seems to agree with tumor DSC and DCE studies. And our estimated GAD concentration is in a reasonable range, as concentration of GAD in blood vessels is typical 1 millimole per liter. And we are still looking for more explanation of these curves parameters. Finally, we did analysis for this whole brain map. We can see the GAD concentration in whole brain like this. The first movie shows all signal we can see. And the second movie shows the signal that we believe is in the brain. And currently, we are working with neurosurgeon to validate with animal model and looking for physiological reason for this signal. For conclusion, we optimized a fast 3D TSC sequence for dynamic GAD-induced MRI change in perivascular space with temporal resolution equals to 9 seconds, spatial resolution equals to 0.75 millimeter isotropic for whole brain. And our initial results in human study demonstrate that our method could detect dynamic signal change after GAD injection in location where cerebral lymphatic vessels are identified. And our method could achieve much higher temporal resolution compared with existing method. And the concentration of GAD in perivascular space is first time being estimated using our method. And our method is also optimized on 70 and could also be used in intracircular injection of GAD study. Thanks for your attention.